Get strong.
Drug assays are a cornerstone of malaria research. It allows scientists to measure how well antimalarial compounds inhibit Plasmodium falciparum growth. These assays help determine drug potency. So, for example, if you look at the inhibitory concentration 50 values or IC₅₀, it also allows you to look at resistance levels and efficacy of new or existing treatments. So we're going to walk through how to do a drug assay, the typical steps that you follow in this video. Choosing between single dose and serial dilution drug assays. In malaria research, two main strategies are used to assess drug efficacy against Plasmodium falciparum. You may use serial dilution assays or single dose assays. In serial dilution assays, you measure parasite growth inhibition across a range of drug concentrations. These generate dose response curves and allow for precise calculation of metrics like IC₅₀ or inhibitory concentration 50. Single-dose assays, on the other hand, involve exposing parasites to one fixed drug concentration. These are ideal for high-throughput screening, field surveillance, or initial triage of compound libraries. Each approach has its place depending on the objective, whether it's discovering new leads, confirming resistance, or comparing parasite lines. So let's look at an example. This example, we're looking at performing serial dilutions in a drug assay. This is in the malarial setting, but it applies in other settings as well. So a typical method for assessing drug sensitivity in P. falciparum is the twofold serial dilution technique, often performed with dihydroartemisinin or DHA or other antimalarials. So the step-by-step example could come across in the literature is first, you synchronize the parasites. So typically ring stage parasites are used. You want 0.5 to 1% parasitemia, and you use a hematocrit or blood cell component of 1.5 to 2%. So you prepare your drug. For example, if you want to use 1 micromolar of DHA, which is like 1,000 nanomolar. So you perform twofold serial dilutions starting at your 1 micromolar or 1,000 nanomolar. So twofold means you just dilute it 1 and 2. So then the next one is 500 nanomolar, 250 nanomolar, 125 nanomolar, 62.5 nanomolar, right down to 1 nanomolar. Then you add your diluted drugs to 96 well microplates containing parasite cultures. So the volume is around 100 to 200 microliters. You incubate the parasite with the drug for 48 to 72 hours at 37 degrees Celsius with appropriate gas mixture. Then you measure the growth inhibition using an assay like Cyber Green 1 fluorescent assay. Then you go ahead and you analyze the fluorescence values and plot dose response curves in software like GraphPad Prism in order to determine the inhibitory concentration 50. And then you can also look at the inhibitory concentration 90. So this method is widely used in both academic and surveillance settings. So hands-on in the lab, you prepare the parasite culture, you synchronize the parasite, again, ring stage. This allows for consistency of readout. So if you have a mixed culture where some parasites are at the trophozoite stage, some at the schizon stage, then you're going to get mixed readouts. So you want to synchronize them to one particular stage, and usually it's the ring stage. So again, you're starting at 0.5 or 1% parasitemia, and you're making your red blood cell component that you add to be, which is referred to as the hematocrit, to be at 1.5 to 2%. So using a 96 well microplate for high throughput, so that you can also have replicates, because you typically want at least 2, you typically want duplicates, but triplicates are ideal. and you are in a total volume of up to 200 microliters, like 100 to 200 microliters. So your drug preparation. So you prepare a serial dilution of the test drug as mentioned across the wide range. Then you're going to add the diluted drugs to the wells. So let's say you have 10 concentrations and each is done in duplicate or triplicate. You want to include a negative control. So this is where you have some of the parasites at the same volume, but you haven't added any drug. So you can reference it and compare it. And then you also want positive controls. You want to use a drug that you know definitely kills the parasite. So for example, if you have a chloroquine sensitive cell line, then chloroquine, or artemisinin if the parasites are sensitive to artemisinin. So that's your positive control. You know that should definitely die. Because if it doesn't die, then you know something went wrong with your assay and you can't deduce any biological meaning from it. So you incubate your plates for 48 to 72 hours at 37 degrees, maintaining the standard gas mixture, which is 5% carbon dioxide, 5% oxygen, and 90% nitrogen. And you don't want to disturb the plates while they are incubating. Then now is the readout. You want to measure the parasite growth in the presence of the drug that you're testing. So you choose a method to assess how much the parasite grew in each well. A common one is to use the cybergreen 1 fluorescence assay. So here you add lysis buffer with cybergreen 1 dye after the incubation. So you incubate it with the dye and the lysis buffer for an hour in the dark. Then you measure the fluorescence with a plate reader. And here you excite the fluorescent molecule at 485 nanometers. And then the emission is usually at 535 nanometers. So you correlate your fluorescence intensity with the amount of parasite DNA, content. Another way that people may read out the parasite growth is to use an isotope of hypoxanthine, where you provide this hypoxanthine isotope, and you observe its incorporation into the parasite, because the parasite actually uses hypoxanthine. So here you add radioactive hypoxanthine at the beginning of the incubation. Then you measure the uptake into parasite DNA using a scintillation counter. So this is quite a sensitive approach, but it does require radioactivity, and so you may not want to handle that if you don't have great procedures in place for safety. An alternative is the lactate dehydrogenase, the parasite lactate dehydrogenase assay. So here you measure parasite lactate dehydrogenase enzyme. This is a colorimetric assay. It's an ELISA-based technique, and it's suitable for non-radioactive workflows. There's also the old-fashioned way of doing microscopy. So you prepare GIMSA or GIMSA stained smears, and you manually count the parasitemia. So this is slow but useful for confirmation. So your data analysis, then what you want to do is plot the dose, so you know what concentration of the drug you had, and then you relate that to the response. And you get these dose response curves, as it's called. So here you have the drug concentration versus the parasite growth. You use curve fitting software such as GraphPad Prism to determine the IC₅₀. So here, this is the concentration that inhibits 50% of parasite growth. Or in addition, you may want to look at the IC₅₀, or effective concentration 50 for other applications. So you compare your inhibitory concentration 50s to standard reference values to assess resistance. So there are also specialized assays. If you are looking at the ring stage, for instance, there's the ring stage survival assay. So here you detect artemisinin resistance by exposing early rings for 6 hours to artemisinin, then washing it off with media. You wash the drug off with media and incubate it for another 66 hours. So the delayed cell death is another approach, another specialized assay you may use, and this is for drugs that target apicoplasts, for example, doxycycline. These effects will be seen in the next parasite cycle. And then there are also things like the checkerboard assay, where you assess drug combinations for synergy or antagonism. So when is only one drug concentration used in research or in your drug studies? So single point drug assays are used in several research and public health contexts. So if you're doing high throughput screening, and we have a video from like years ago on this page, on this channel, talking about high throughput screening and how it's done. So in this setting, you're looking at thousands of compounds that are tested at one fixed dose, for example, 1 micromolar. And from these sort of assays, hits are compounds that show high inhibition, so greater than 80%. There are also resistance surveillance assays where you may want to just pick just one dose. So here with the ring stage assay, survival assay, for instance, you use a fixed dose of, for example, 700 nanomolar dihydroartemisinin. And these are often used to detect field resistance trends with fixed drug panels. Then in functional genomics and phenotyping, you may just pick one single dose to assess whether your gene knockout or mutant lines are sensitive or insensitive to a drug that you're testing. So here your gene knockout or mutant lines tested at one known inhibitory dose, for example, at an IC₅₀ dose. And here you use it to compare relative survival or resistance. So single-dose assays are time-efficient and cost-effective, especially for large-scale studies or preliminary screens. Thank you for joining me on this video. If you enjoyed it, you may also want to check out the parasite life cycle, which has already been covered, or you may also want to check out a run through of Plasmodium as a group in general. They're very unique creatures. So thank you once again. Please subscribe, give it a thumbs up if it was helpful, and I will catch you in the next one. God bless you.